Melatonin, the hormone of darkness, which is the unofficial title given to melatonin by one of the researchers whose articles we're citing. And we will be presenting on melatonin. So its official chemical name is N-acetyl 5-methoxytryptamine. It's a hormone, more specifically an indolamine, which is it's a monoamine neurotransmitter. And melatonin bears a lot of similarities to serotonin and tryptophan, and this makes sense because serotonin and melatonin are both the type of hormones that can be synthesized from amino acids. Okay, and melatonin is a derivative of serotonin, so we'll be talking more about that in a little bit. In terms of what organisms produce melatonin, the answer is pretty much everything. Bacteria, unicellular eukaryotic organisms, invertebrates and vertebrates, algae, plants, and fungi all produce melatonin, and that's according to one of our researchers, Carrillo Vico. And there's also, there has been research done that found melatonin in alpha proteobacteria and cyanobacteria. And a hypothesis about the reason for this, because it's clear that melatonin developed early in the evolution of these organisms. One theory is that it's because it's such an effective antioxidant, so it helped preserve the health of all of these organisms in their early stages, especially in an environment that was heavily exposed to UV radiation. And that is according to Slaminsky and his fellow researchers. So melatonin is primarily synthesized in the pineal gland, but it is also synthesized and secreted in the brain, lungs, liver, kidney, carotid body, reproductive tract, skin, and more. And there are some interesting implications of this that we'll be talking about in a little bit. This is again according to Slaminsky. There are two receptors for melatonin. Both are G-protein coupled receptors. MT1 controls neuronal firing and metabolism, and MT2 controls the circadian rhythm, vasodilation, and the immune response that melatonin corresponds to. So the responsiveness of these receptors is determined by a number of factors, but that includes the circadian rhythm, the amount and duration of exposure to melatonin, and whether the melatonin that the receptor is exposed to was created in the organism or introduced from a foreign environment. This according to Dubokovich et al. So melatonin doesn't just have an effect on the organismal level, it also has an effect on the cellular level, which makes sense, but we primarily hear about it as it affects the body as a whole. On the cellular level, melatonin has an effect on the endoplasmic reticulum stress response, the regulation of apoptosis and autophagy, mitochondrial homeostasis, and cellular homeostasis. And a lot of the effects that it has on the organism as a whole, it also has on the cellular level, helping to stabilize responses to things and preserve the health of the cell as a whole, according to Hardland et al. So melatonin influences many areas of the organism as a whole, and primarily we hear about it as it pertains to the circadian rhythm, but it also is involved in bone formation and protection, reproduction, cardiovascular health, gastrointestinal function, psychiatric disorders, and there's ongoing research investigating a therapeutic role of melatonin in psychiatric disorders, cardiovascular disease, and oncostatic effects. And one of the reasons that melatonin is so good at combating various types of disease is that it's a powerful antioxidant. We'll be talking about that in a little bit. But in terms of oncostatic disease, which is cancer, melatonin has been administered along with chemotherapy to aid in the health of the patient, because chemotherapy often induces the production of free radicals, which can cause a lot of damage to the person. But melatonin, because it's such a strong antioxidant, combats some of those effects. This according to Torsman et al. So one of the functions of melatonin is as an immunoregulator, and that's well researched. And there are various theories about what exactly melatonin is doing and which response it primarily controls. One is that it's an anti-inflammatory, one is that it's primarily an immunostimulant. But there's another theory that melatonin is an immune buffer, acting both as an immunostimulant and as an anti-inflammatory under various circumstances and controlling different responses. There are strong links between the neuroendocrine system and the immune system. Both systems act on each other and make use of a number of the same types of compounds, or the exact same compounds that are shared between the two systems. The pineal gland is part of the neuroendocrine system, but the presence of melatonin and the enzymes that are involved in its synthesis are found in areas all over the body that are not part of the neuroendocrine system. And this, in conjunction with the link between the neuroendocrine system and the immune system, is strong evidence that melatonin is involved in the immune response in one way or another. In addition to the studies that have been done showing the immune response and how it's affected by greater or lesser presence of melatonin and other such factors that have been studied. There's also an age-associated decline of melatonin that's linked with an age-associated impairment of the immune system. And as melatonin production declines, the overall health of the organism declines. And this also occurs in the brain, where lower levels of melatonin tend to lead to higher levels of brain disease, again, because of the antioxidant properties of melatonin. And this is from Carrillo-Vico et al.
So, in order to talk about melatonin and its main role in the circadian rhythm, we need to go over some anatomy and physiology and the effects that melatonin has on the macro level before we can dive into the micro level. So first, the circadian rhythm. A lot of people associate with sleep regulation, which is a huge part of the circadian rhythm, but the circadian rhythm monitors other things besides sleep regulation. It monitors temperature regulation of the body, when to raise and lower it, so depending on season changes as well. When you're supposed to eat or not, like your body develops this internal cycle and regulation and schedule of your bodily activities and what you need to do throughout the day. And it's kept within your brain in the hypothalamus. And this certain area that regulates this is the suprachiasmatic nucleus. From AMP, the nuclei are the cell bodies associated together of neurons. So that particular nucleus is what regulates the different cycles within the circadian rhythm. So the sleeping, the eating, even socializing, as mentioned in the study from the British Journal of Pharmacology from Nava Disapel. And one of the big things that this researcher also hit on is that it's light regulated, meaning that the circadian rhythm and the release of the different hormones within this is depending on when it's light out or when it's night, and this forms the circadian clock. So with melatonin, as we'll see in a minute, it's going to spike towards this moment known as deep sleep. And it's because melatonin is what activates your body into going into sleep mode, which the body needs in order to preserve energy in the brain. We'll see this in a minute. Another key factor of the circadian rhythm is that this is the reason why it's light regulated is because it's due to wavelength sensitivity. The eye receptors in the different cones and rods as seen, as well as just wavelengths that penetrate from photopigments, can determine what is released at what point in hormones as well. So the one part of the circadian rhythm is the sleep-wake cycle, which most people associate with the circadian rhythm. So this is induced and regulated by melatonin levels and also another hormone called cortisol, which is the stressor hormone that wakes you up in the morning. Melatonin ends up spiking towards the end of the day when the sun starts going down because of this light dependency. Now, the need for sleep is seen throughout the brain physiologically because the brain has to have a moment of restoration. It needs to be able to replenish the energy that it used up during the day. It needs to process and remember all that it has collected within that day because so much has happened in such a long period of time, and it needs a moment to recollect without doing very minimal things, even though it is still doing a lot of things, according, again, to the same researcher, Nava Zispel. So melatonin secretion. The way melatonin secretion occurs is first the suprachiasmatic nucleus sends out a neuronal impulse once the tracts within the eyes take in certain light waves that tell the body that it's night. The nucleus sends out a signal. The signal reaches the pineal gland, and melatonin is released and is synthesized, and we'll cover how it's synthesized on a micro scale later with our mechanisms. It is released within the third ventricle, which is this opening within the brain that can reach different parts of the brain, as shown by the figure. And then it enters into circulation. So the body starts hitting this moment where it's turning into this sleeper mode, and the brain starts hitting this network called the sleep default mode network, which is the parts of the brain that are actually working during the night while you are sleeping. So certain parts of the brain do shut down during the night, but certain parts of the brain have to be working during the night or else be classified as being alive. Again, this was from the same researcher, Nava Zispel. And you can think about the sleep-wake cycle and the role of melatonin, as well as understanding light regulation from real-world problems we have all probably experienced. One being jet lag. We remember possibly traveling a far distances for a short amount of time, maybe 7 days or so, a week even, and not being used to waking up and going to bed as saying in a foreign country as opposed to here in the States, because our brain is so adjusted to waking and sleeping at a certain time, and it's working overtime to try to adjust to it because it's already set in this routine. That's what the circadian rhythm is. It's a routine. Basically, your brain has set aside for you. And that's another reason why daylight savings time is a hard thing for some people as well, because your body is so used to releasing melatonin at a certain point that when it's either light out during earlier than usual, or it becomes dark out later on, your body has to work a little bit harder to try to adjust to that change.
So melatonin's biosynthesis begins with the amino acid tryptophan, which is converted to 5-hydroxytryptophan catalyzed by the enzyme tryptophan hydroxylase, which uses a tetrahydrobiopterin cofactor, as well as oxygen. 5-hydroxytryptophan is then converted to serotonin through an enzyme hydroxytryptophan decarboxylase, which uses a pyridoxal phosphate cofactor and releases CO2. Serotonin is then converted into N-acetylserotonin by an enzyme called serotonin N-acetyltransferase, using acetyl-CoA and releasing coenzyme A. N-acetylserotonin is converted into melatonin by the enzyme hydroxyindole O-methyltransferase, which uses S-adenosylmethionine and converts that into S-adenosylhomocysteine. This is according to Bayer et al.
So next is the tryptophan hydroxylase. So we start with a cofactor known as tetrahydrobiopterin, oxygen, and then iron 2+. So first we start off with a deprotonation of the tetrahydrobiopterin next to the carbonyl, deprotonation of that nitrogen, and the electrons move and they pass it along the different double bonds as shown. And the last double bond attacks the oxygen, displaces the double bond between the oxygens to attach to the iron. And then within the next step, the iron's electrons displace onto the oxygen it's attached to, which then releases that one oxygen to be protonated to form an alcohol, or to form an alcohol with the tetrahydrobiopterin, And so that then turns the iron from a 2+ into a 4+ because the electrons move to form that double bond. So you lost 2 electrons by forming that double bond. So then tryptophan comes in, and the double bond off of tryptophan attacks that oxygen, and that displaces the double bond from the oxygen and the iron, 4+. And then this attaches the oxygen and the iron to the tryptophan. Then the displacement of the bonds with an iron are displaced down to form the carbonyl back on the oxygen. And then there's a hydride shift on the carbocation that was formed from the alkene that attacked the oxygen in the previous step. Then a base comes in, attacks one of the hydrogens, that is displaced, forming a double bond, and then the carbonyl is then displaced to attack a proton, forming an alcohol. And this forms 5-hydroxytryptophan. And this is from Roberts et al.
The next step in melatonin biosynthesis is catalyzed by the enzyme hydroxytryptophan decarboxylase, which I could not find a source to cite the mechanism for. But since it is a decarboxylation reaction using a pyridoxal phosphate cofactor, I'm suggesting this mechanism, proposing this mechanism, which begins with 5-hydroxytryptophan forming a Schiff base with a pyridoxal phosphate cofactor. It begins with the nitrogen getting deprotonated and then attacking the aldehyde on the pyridoxal phosphate. The electrons collapse down from the nitrogen to form a double bond with the carbon, and the hydroxide on the carbon is lost. The carbon dioxide then leaves, pushing electrons up to form a double bond with the nitrogen, which pushes electrons along, moving the double bonds around the ring, and finally pushing electrons onto the nitrogen on the pyridoxal phosphate. The nitrogens then jump back to reform a double bond on the other side of the pyridoxal phosphate, and the electrons are pushed up back onto the double bond between the carbon of the pyridoxal phosphate and the nitrogen, and the electrons from the double bond attack a proton. Hydroxide then comes back to break the Schiff base, attacking the carbon that was originally the aldehyde of the pyridoxal phosphate. The double bond electrons between the nitrogen and the carbon are protonated, attack a proton, and a base comes in and deprotonates the alcohol, reforming the aldehyde and releasing serotonin.
So the last mechanism we'll be talking about is serotonin N-acetyltransferase, also known as SNAT. And this is from Molecular Cell, written by Hickman et al. So the mechanism starts with serotonin. The amine is deprotonated by a base, and the bond to the nitrogen then goes and attacks acetyl-CoA, pushing the electrons up onto the oxygen of the carbonyl. This gives rise to an intermediate where the amine is bound to acetyl-CoA. The electrons fall back down on the oxygen, pushing off acetyl-CoA and giving us another intermediate where the base comes in, deprotonates the amine, pushing the electrons back down onto the positively charged nitrogen, and giving rise to an acetylserotonin, where you have the carbonyl group and the deprotonated amine now.
So, for the regulation of melatonin biosynthesis, early studies suggested the enzyme hydroxyindole O-methyltransferase was the rate-limiting step. However, later studies identified serotonin N-acetyltransferase, also known as SNAT, as a key regulatory enzyme. SNAT is regulated by norepinephrine, which binds to a beta-adrenergic receptor, which activates adenylate cyclase, which produces cyclic AMP. Cyclic AMP can then bind to the regulatory subunits of protein kinase A, causing them to fall off and activating protein kinase A. Protein kinase A can then phosphorylate SNAT, and this phosphorylation allows an interaction between the phosphorylated SNAT and regulatory 14-3-3 proteins. This interaction with the 14-3-3 regulatory proteins stabilizes SNAT and prevents its degradation by the proteasome. And this is important because SNAT activity is determined by the amount of enzyme. And that is according to Chalmers et al. So in primates, SNAT is constantly synthesized, and the amount of the activity is determined by the amount of the protein, which is determined by how much is degraded, and that is stabilized at night by protein kinase A phosphorylation. In rodents, SNAT transcription is activated by cyclic AMP, response element binding proteins, also known as CREBs. CREBs are activated by phosphorylation by protein kinase A. Protein kinase A was activated by cyclic AMP from the norepinephrine signal. So it's a very similar type of regulation, and then it goes through cyclic AMP and protein kinase A. But in primates, there is a constant level of transcription of SNAT, and the degradation of SNAT is regulated by that pathway. In rodents, transcription itself is regulated by that pathway, going through norepinephrine, cyclic AMP, and protein kinase A. This is also according to Chalmers et al. However, later evidence was gathered against SNAT as the rate-limiting enzyme in melatonin biosynthesis. Researchers discovered that rats with mutations reducing SNAT by tenfold showed normal circadian rhythm and pretty high regular melatonin amounts, which suggests that SNAT is not the rate-limiting enzyme for melatonin. Other researchers also found that melatonin levels did not rise and fall with SNAT levels. So the hypothesis shifted back to HIOMT as the rate-limiting enzyme for melatonin biosynthesis, and that is according to Tan et al. Other researchers found that patients with autism spectrum disorder who also had HIOMT mutations showed decreased melatonin production, which provided further evidence that HIOMT is a rate-limiting enzyme for melatonin synthesis. This is according to Leo et al. The current theory of melatonin regulation is that SNAT is the melatonin rhythm-generating enzyme. The surge in SNAT at night, when it's no longer degraded through the signaling pathway previously discussed, drives the daily rhythm of melatonin production. So during the day, low levels of SNAT limit melatonin biosynthesis because the SNAT is degraded very quickly by the proteasome. At night, however, SNAT levels rise as degradation is prevented by phosphorylation of SNAT. So HIOMT is the rate-limiting enzyme. This is according to Leo et al.
So melatonin can act as an antioxidant and can act like one too. It has an electron-rich aromatic indole ring that can donate electrons to other radicals. It can be a better antioxidant than vitamin E and C, and it does not undergo redox recycling, so it's irreversibly oxidized, and that means that it is a terminal or suicidal antioxidant. That means that you can't go back and reuse it over and over again. The body has to synthesize new melatonin for antioxidants. It's important for the brain's oxygen-rich environment. So again, as we mentioned before, melatonin, and along with the rest of the circadian rhythm, is very important to homeostasis within the brain. And this is from Tan et al. So melatonin enhances other antioxidation mechanisms, as proven from the studies from Tan et al. It enhances the activities of other antioxidants, which the studies show that vitamin C and E showed an increase in antioxidant activity when combined with melatonin. This stimulates the synthesis of antioxidants. For example, one that showed up was glutathione, and it stimulates antioxidant enzymes like superoxide dismutases and catalases, and it also suppresses radicals generating enzymes. The example is it downregulates lipoxygenases. And again, this is by Tan et al.